Good day, this is Professor Will, CBMD PhD. Today is Wednesday, September 30th, 2020. It is 6:02 and 23 seconds p.m. Eastern Standard Time. And this is a continuation of my microbiological readout. So let's hop to it here. The eubacteria are the consummate metabolists, sheer metabolic power and versatility. They are the masters of metabolic theme and variation, just as eukaryotes are masters of structural theme and variation. Someday we will understand just how eubacteria could have acquired such a varied metabolic repertoire. Their versatility is the more interesting when one realizes from genomic analysis that much of the more limited metabolic repertoire of the archaea, beyond that characteristic of the methanogens and methanogenesis, is, as alluded to above, the product of metabolic capabilities, enzymes, and pathways imported from the eubacterial world. The phylogenetic structure of the eubacteria is rather different from the archaeal one just discussed. Viewed from afar, the main eubacterial lineages appear to radiate out from a crown group, with a few atavistic forerunners branching from the eubacterial stem slightly earlier, examples being the aquificals and the thermotogales. It is the crown radiation that spawns the phenotypic richness we characteristically associate with the eubacteria. Among the major kingdom-level eubacterial taxa, those dominant by virtue of numbers, phylogenetic diversity, and ecological impact, there exist several kingdoms of phyla about which we know next to nothing. Only a few, if any, cultured species represent them their evolutionary breadth and ecological prominence being demonstrated by large numbers of environmentally derived rRNA sequences from the various groupings. The lesson that has yet to sink in is that microbiologists know relatively little about the bacterial world. We clearly have no useful idea of what some of the major environmentally important bacterial groups are and are doing. You would think that a great deal of work would now be going into remediating this situation. This is not the case. Let us begin with the major, most significant eubacterial kingdoms and phylae, and then, when appropriate, extend to their subordinate classes. I more or less lump kingdoms and phylae here, because until the proper genomic criteria have been developed to define taxonomic rank on the basis of the properties of naturally defined organismal groups, it is not, in some cases, possible to distinguish the two. The Proteobacteria, one of the most, if not the most ecologically dominant eubacterial kingdoms, is the Proteobacteria. The kingdom comprises 5 phylae distinguished by the Greek letters alpha and epsilon. The alpha, beta, and gamma phylae obviously cluster, which easily emerges by a number of different phylogenetic analyses, while sigma and epsilon, which may be specifically related to, stand apart, but still within the Proteobacteria kingdom. The beta phylum is actually a major branching within the gamma. I still consider beta to be a phylum rank because of the apparent phenotypic diversity within it, and its branching from a section of the gamma tree, which except for a beta branch contains no photosynthetic representatives. A taxonomic precedent for such a ranking is given by the class of mammals, arising from within the class of reptiles. The alpha phylum, the phototrophic alpha of Proteobacteria, to a first approximation comprises purple, photosynthetic lineages, non-photosynthetic derivatives thereof, and a variety of pathogenic groups, mainly intracellular pathogens. The most notable of the latter is the Rickettsia group, the order Rickettsia, which comprises the genera Rickettsia, Wilbachia, Ehrlichia, Anaplasma, and Cadria. All which seem to be free-living relatives of these pathogens have been detected by rRNA analysis from marine habitats, although none have been cultured. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and computer science. I hope you enjoyed listening. Until next time, have a great day.